Hi, everybody. Welcome to our monthly webinars. I'm Mala Kumar, a freelance writer and a consulting editor at iwonder. iwonder is a science magazine for middle and high school teachers. It features writings about the many dimensions of teaching and learning of science in class and outside it. This magazine is published in English, Hindi, and Kannada twice a year. You can read and download our latest issue from our website. If you are a practicing science teacher, we invite you to write for us. To do this, please send in your idea in 100 words to iwonder@apu.edu.in. If you're looking for themes to write on, we share one at the end of each webinar. There are three questions at the end of the August webinar, and there's one at the end of this one. If you'd like to receive updates of each webinar and a free print version of each issue, do subscribe to us by following the link displayed in the description of this video. You can also catch us on Facebook. As you may know, on the second Wednesday of every month, we invite authors of an article in iWonder for a live discussion. For this webinar, we are delighted to bring back our very first guest, Rohini Karandikar. Rohini, thank you for being with us today.
Thank you, Mala, and I would like to thank the entire iWonder team. I'm honored to be a speaker at this webinar for the second time. And I also want to congratulate the iWonder team for entering their fourth year of this insightful webinar series. I'm looking forward to a lively interaction.
Thank you. Thank you, Rohini. Rohini is a science educator with a PhD in biotechnology from the Indian Institute of Technology, Bombay. Presently, she works as a QA consultant with an ed tech company, Hubble Hawks Technologies. Rohini also writes in English and Marathi on popular science topics and on education. Our discussion today will be around her article, A Milky Way to Learn Biology, published in the August 2019 issue of I Wonder. Before we start, let me give our audience a brief overview of the format of the discussion. For the first 30 minutes, I will ask Rohini some questions related to her article. We request you, dear viewers, to type your questions in the chat box. In the last 30 minutes, we will request Rohini to respond to your questions. Now, we are right here and we are going to start. Rohini, all set?
Yes.
Okay. Rohini, setting curd is one of the most common and routine activities that children, irrespective of their background, may have seen in their homes. Yet, this is the first time we have come across a science learning module designed around this activity. How did you think of this?
This module is based on a project called Vigyan Pratibha, or VP, we call it. Vigyan Pratibha is an initiative by the government of India. And in 2017, it was assigned to the Homi Bhabha Center for Science Education, that is HBCSE. And I was working at HBCSE as a visiting fellow. So we were asked to design activity-based modules for high school students from diverse backgrounds, and
Mhm.
with the aim of nurturing their talent in science and maths. So the modules had to be based on using simple material, low cost resources, or easily available material.
Mhm.
So we used, as in different teams, physics, chemistry, maths, biology, we used different resources like marbles or graph paper, grid paper, carton boxes, and we designed different modules on different topics. And curd formation was one of those modules. So
Okay.
that was how it started. And
Okay.
I also want to mention that while we were working on this module, we also visited some schools where we tried out this activity with school students in their laboratories or their classrooms. Similarly, we also tried these activities at summer camps or at camps at HBCSE. So we engaged teachers and students both in these activities before it became a full-fledged module.
Okay. Okay. I can imagine the fun the kids must have had. Uh, what were your aims at designing this particular module around this activity? designing a module around this activity, I mean?
Uh, First was to introduce students to the idea that curd formation is a scientific process, and it is based on the principles of physics, chemistry, and biology. Secondly, we wanted to take them or introduce them to the scientific method, about which I'll also be telling in today's session. Like how does one think of an experiment? How does one design an experiment? And all those aspects of the scientific method. And we also wanted to tell them that biological processes take time. So you might work on a physics activity or a maths activity for 2 hours and it's done, but that's not the same for biological processes. It depends on which organism you're using, how fast that organism grows, how does it behave, and what aspect of its growth or its behavior are you looking at. So it takes heaps of patience, but it's fun to work in biology.
Okay, good. Uh, Many experiments at the school level are aimed at exploring a single topic or theme in the curriculum more deeply. You use an unusual approach. You start with the simple activity of setting curd and show how it connects the practice of science with many concepts from different topics and different subjects in the curriculum. Could you very briefly share some of these concepts?
Yes, we connected this activity with a number of concepts. Like in CBSE, they have a topic on microorganisms, friend and foe. So, where they introduce how not all organisms are bad, most of them are very good, some of them are very helpful to us.
Mhm.
That is one concept where we talk about beneficial microorganisms and their role in our life. It also connects with the concept of acids and bases, because curd formation is a process where uh, lactic acid bacteria ferment lactose to lactic acid. So we don't talk about these things we right initially, but we know it is based on the acids base concept. We also talk about the effect of temperature, because students know that curd formation has to be done at a certain temperature. It takes time during winters, but it happens quickly in summers. So how does the environment affect this biological process? That was the other point we wanted to connect it with.
Okay. So, between these two approaches, how does this difference in approach influence the nature of learning?
I think the way it is taught, or the way students learn from a textbook, first of all, it appears to be a bit technical, because when we ask them questions about these topics, they tend to sort of repeat what is there in the books. But when we come to this activity, it is
Hmm.
mainly based on their experience, especially when we talk about it for the first time. They will tell
Hmm.
what they have seen or what they know. If they have tried it at home, they'll tell how it felt doing it. So it comes from their experience rather than textbook knowledge, or rather than them using very technical words to explain what they know.
Okay, okay, right, that's true. Um, you suggest that children can learn concepts better when they do science, you know? So, what does doing science mean?
In this case, I'll say doing science is where they are using their knowledge, their prior knowledge, along with critical thinking and asking a lot of questions,
Hmm.
curiosity. So they are using all of these to try to answer, or try to solve a problem. So in this case, broadly, the problem is tell us a way to set curd. And then they think about different conditions. They think about the volume of milk. They think about what should be added, or should something even be added to milk. They think about all these things. So they
Hmm.
are doing science in a way that they are first thinking about what they know and combining it with the other things like critical thinking, questioning,
Hmm.
and even predicting, like, okay, this will happen or this will not happen. So they do that a
Hmm.
lot.
Um, and you suggest that one advantage of doing science is that it gives students the opportunity to experience what it means to be a scientist. Is that important? Why is it important for them to feel like a scientist?
Um, So most educators will agree that the way practicals are done in school, or the way students work on an experiment, is in a very cookbook style, where
Mm.
students write the aim, objectives, materials and methods, procedure, everything is properly, nicely written even before they see the apparatus. And then they have to follow a certain method. It's again, it's written, your ABCD, and then they know what is to be inferred, what should be the answer, or not really the answer, but it has to be written in a certain way.
Mm.
Whereas the way a scientist works is very different. First, a problem is identified, its relevance is also like discussed, whether it is relevant, and
Mm.
then a hypothesis is formulated. Then scientists design an experiment. Then they observe, they record their observations, and they infer or they draw conclusions only on the basis of what they observe. So there is nothing like, you have to get this as the answer. How many of you have got this? Which usually happens in classroom interactions.
Correct.
So it's very different than that.
Mm.
So also many times scientists get very different results than what they expected. So
Mm.
then you have to go back to, reformulate the hypothesis again, design a new experiment, work on that. Even if they get an expected or a desired result, the experiment has to be repeated to show that the results were not obtained by fluke.
Mm.
So that is another thing, and then we have to show that the experiment or the results are statistically significant. So it has to be repeated a number of times. At times even peers have to repeat the experiments to ensure that it is reproducible, the experiment is reproducible, and they are also getting the same data or similar results. Then it is discussed, it is peer reviewed, and then it is published. So scientists do a lot more than this, but the scientific method is the same, where it is difficult right at the beginning to tell that, okay, this is going to be the result of my experiment, or these are the steps I'm going to follow, I'm not going to make any changes. That is very difficult for a scientist to do.
Mm. So, from different results to different images, my next question is about teachers and students. All of us may have, we may hold different images of what it means to be a scientist. What aspects of this experience did you have in mind while designing this module?
While designing the module, I had not thought about how students perceive a scientist, because
Mhm.
it was my initial, I was just venturing into education. So the trials of this activity were my first trials in a schools within a school setting.
Mhm.
But when we asked students about being a scientist, or what they how they view a scientist,
Mhm.
I had a very I'll say a very predictable experience. So I had gone to a school with one of my colleagues, and we both happened to be scientists previously before we joined HBCSE. So when we told students that we have been scientists, they looked at us in disbelief. It was very difficult, and we saw, I still remember the faces. It was very difficult, and that's also very natural. And why I said predictable is because that's how media portrays scientists. Their perception is scientists wear a lab coat and they carry equipments, they have some laboratory apparatus in their hands, they're always thinking, they're busy, they live a very lonely life. That is what in general many students thought about. But yes, and I think that is also because the interaction of scientists with students is not to the level as it should happen. So that also shows that scientists need to interact more with students, and even
Mhm.
more with students from the remote areas, because they are not likely to have seen a scientist. So
So, we
there again,
really
sorry.
carry on, please. No, carry on,
Uh,
carry on, Rohini.
yeah, also the topics of research, when we talk to students about scientists, not just in this activity, for other activities also, students mainly think about topics like cancer research, like scientists work on cancer research, finding a cure, diagnosing cancer, or vaccines for malaria, AIDS, or space research. But
Mhm.
what many people, not just students, many people are unaware of is that even the fundamental topics in science are still ongoing as research topics. We are not yet done with many fundamental topics in science.
Hmm.
So that is one surprising part of this.
So I was just saying that we're really happy to have you, the scientist, here on our webinar.
Thank
Um,
you.
Often we give students the kind of experiments scientists do and the kind of questions that scientists ask. You use a different approach. You use an activity that is common to our homes and raise questions that anyone could ask. You then invite students to explore how scientists would respond to these questions. Could you elaborate on this difference? What advantages does this offer?
When we start with okay, before we give the activity to the students, before we thought of this as a topic, we wanted to give students something that they are not very like too familiar about, but at the same time, it should not be something that they don't know anything about, because that would have taken a lot of time in researching and reading about it. So for example, in this case, most students know that for curd formation you need milk, and you probably also need something else. But what happens in bit what happens when you add that something to the end step of curd formation? That is something students don't know very clearly. There are mixed responses from students. They debate amongst themselves, no, it happens like this, no, you have to do it like that. And then so it's through this activity we make them look at the whole process in real time. So generally what happens in homes is that you add curd to milk and you keep it for like, oh, you keep it overnight. But what's happening in between, we don't check really. So here what we tell them is, okay, this is a process you're all familiar with, but you have to investigate this process. Okay, do you know how much curd has to be added? What should be the proportion of curd is to milk? At what temperature should milk be before you add curd? Where should you keep the whole experimental setup so that you get the curd in little time, in lesser time? When do you say that curd is ready and now I should keep the pot in the fridge? So where to stop that experiment? So these are the aspects we thought students can think about, and then we say, okay, investigate this process like a scientist does. So I think that is what gets students curious about the process.
Lovely. Rohini, you suggest that another advantage of doing science is that it helps children develop a broad understanding of the scientific method. Since there are many variations of the scientific method, could you briefly outline which aspects of this method you focus on in this module and why?
We focused on hypothesizing, experimental designs, um, variables, like what variables to choose and how to vary those components, observation and data recording, and drawing conclusions or inferences. But we didn't explicitly use these terms that, okay, you are now formulating a hypothesis or you are now drawing a conclusion. We didn't use these terms
Hmm.
in the students' sheets.
Okay. Um, so, teachers who want to try this module in class may have two options. One is to introduce the scientific method, then share your module. The other is to share your module and use what students do in this module to introduce key aspects of scientific methods. Method of which of this would you recommend?
I would recommend introducing the module first and then leaving the students to do the activity.
Hmm. Okay, so
The
module.
reason is because many students do not know, like in our experience, many students didn't know about the process at all. For example, we also received responses like, you just take a cup of milk and leave it outside and it'll form curd. So
Okay.
it'll be helpful to first introduce a module and then allow students to work.
Mhm.
For this question, I also want to tell another aspect, that is about time. Time is a big like it was a bottleneck when we tried it in schools, because I agree that teachers get just one or two, they may get one or two free periods for doing such activities, and one can do only so much in like an hour. So what we suggested and what probably worked out was that teachers would introduce the module to students in the class, students would try it at home,
Mhm.
and then the next they would record, and next day morning they would get the results or their observations to the class, and then
Mhm.
there can be a discussion on what they observed.
Mhm.
So that's another way to do it. It can be chunked into different sections and be done.
That's That's very useful. I'm sure teachers can try that out that way. Some teachers may also wonder if the two advantages you highlight, an introduction to the scientific method and the experience of being a scientist, can be separated from each other in practice. How would you respond to that?
I agree that it'll be very, or rather, I would say students do not easily accept that it is like being a scientist while working on this activity is like how scientists work. Initially, they find it very difficult to accept, which is again a very natural thing because they may not be aware of the scientific method. But then, isn't the scientific method something that we are all using always? Without saying or without telling or without knowing? For some people they may not know, but they are actually using the scientific method. So
Mhm.
we give different examples. For example, I am fixing a broken clock. So
Mhm.
what should I first do? I'll probably first try changing the batteries. If that change also doesn't work, maybe there's something wrong with the spring, then I'll check the springs. If that also doesn't work, I'll check the wires. Are the wires properly connected? Are the wires damaged? Are the hands working properly? So we are using a scientific method in solving so many problems. Or take the example of cooking. I'm trying to cook biryani using my friend's recipe, and I'm not able to nail that taste. So I'll call and ask, okay, what's going wrong here? I don't know. And then she'll say, first you try changing, try using powdered cinnamon instead of the whole cinnamon stick. And then I say, no, I'm still not getting it. Okay, maybe use whole peppers instead of powdered pepper. So I am using the scientific method. So
Right.
that is what, for students, immediately, even if they do not accept, with more and more evidence, they accept this fact that, okay, this is also scientific method. And I am, so for a part of the activity, I mean, scientists do definitely do more than what just the scientific method. But for part of this activity, I am following the scientific method. Eventually they realize.
Okay. Now let's come to the activities. In the first activity of the module, students are asked to interview family members and neighbors about how they set curd. Some may argue that it would be simpler to ask students to observe how curd is set in their own home. Why did you choose interviews instead? What advantages do you think this approach offers?
Observations can be quite passive, and not all students may actively observe. They may just observe and come back without even asking questions.
Hmm.
Whereas when they are interviewing someone, they know that they have to carry back a narrative or a story which they can share with their friends or teachers. So they will be more likely to ask a lot of questions, and beyond what questions we have suggested, they might also add a few questions from their side. So that is one reason we thought interviews would be better than just observation.
Okay, great. That's a beautiful way of presenting it, Rohini. Um, Before students start their interviews, you suggest introducing them to the idea of a science reporter. What does this mean and why is this important?
Again, many educators will agree that role play is a powerful learning technique, and students like doing it.
Hmm.
So we thought if students step into the shoes of a reporter, they will probably, try to act like a reporter, ask questions in that style, and they'll try to get as much information as possible.
Hmm.
So that is one major reason we thought we'll ask them to be a journalist and then ask questions. Again, some of them are aware how journalists question their respondents. They are
Hmm.
not biased. They will not react, but they can ask follow up questions. So students try to do that as well.
Okay, okay. Now, like journalists, they'll also get a lot of answers, huh? So students may receive some conflicting responses from their interviews. How do you help make sense of the information they gather?
At this point, I feel it'll be enough to get responses from their respondents, some answers
Hmm.
from the respondents. Of course, they should be discouraged from flagging any response or judging any response. They can
Hmm.
ask follow up questions, like I said, and eventually, students are going to compare their responses with their friends. and they are going to find commonalities in the responses. So the aim of this task is just to get information from their parents or grandparents or neighbors. So as long as that is done, it shouldn't be a problem.
Okay. This gives a beautiful image, of family and friends all acting and giving questions and answers and all that. You share that this activity can be used to discuss science society relationships. What does this mean and why is this important?
um, Curd, considering curd, it
Mm,
has applications not just as a food, it is also used in many religious practices, and it is also used as a cosmetic.
mm,
So considering this wide application of one food, that is curd, if you just look at curd from the point of view of food, again it has so many applications, uses, right from chutneys, raitas, curry, to salad dressings, or even marinade for meat. So depending on why, for what purpose is someone using curd, the same kitchen will have a different, or the same family will have a different way of preparing curd. So it's not just across different cultures within the same community. Different families or the same family will make it in different ways. Again, people prefer thick,
mm,
creamy curd versus some people prefer very smooth and milky version. And some homes they prefer a mildly sweet version, whereas in other families they prefer a slightly tangy version. So what is it you're using curd for? That determines how it is going to be prepared by the same person again. So with such a
right.
wide, such a diversity, it'll be good if students acknowledge this part, that okay, curd is used for different reasons in different places, people consume it in different ways, and for different reasons again. So I think that's the main idea behind this.
My God, that's a lot of lot of information and for the children to gather. One question you ask students is: are people with no professional training in science also capable of engaging with the scientific method in everyday lives? What kind of responses did you receive?
Initially, most students said no.
Hmm.
Again, because of their experience or their understanding that whatever we are, like, most of the things we are doing is following scientific method. But once they think from that point of view, that okay, solving a problem is also a way of scientific method, or cooking something for the first time is where you use the scientific method unknowingly. Once they get more evidence or they get this idea better, then they appreciate this fact. But initially, no, they failed to, they disagree.
Okay, okay. Your article shows that curve setting may involve many aspects of scientific method. But many of us set curd as we have been instructed to, or have seen others do it, without actually inquiring into or understanding the science of the process. Would it be important to address what role following traditions without personal inquiry has in the practice of science?
It's very important to ask questions. For example, my mother, when she taught me how to set curd, she said you have to add a spoonful of curd to, say, a cup of milk, and then you have to stir it 100 times. And she emphasized on the number 100, and I asked her, why 100, why not 50 or 150? So the
Hmm.
idea is that it has to be uniformly mixed, because curd is very, thick at times, it's very thick, and then when you mix it, it might just remain as that clump, it will not get uniformly mixed. So that is the idea. But when anybody asks a lot of questions on these traditions, they will get some answer. Now those answers can be unscientific or they can be scientific, but that can be decided later. But it is very important to ask the question in the first place: why do we do this? And I
Hmm.
think as students develop the way of scientific thinking, they will also be able to find out the correct answers.
Right, right, right. Okay, so I'm very glad I'm asking you question after question. I'm being a scientist. You talk about how the concept of hypothesis could be introduced through this step. Could you explain this with an example?
In simple terms, a hypothesis is an explanation to a process which I am yet to investigate. So in this case, my hypothesis, suppose I'm new to curd formation, I don't know how it is formed. My hypothesis can be something as simple as, take a cup of milk and squeeze half a lemon juice into it, the juice of half a lemon into it, and leave
Hmm.
it. And that's how you'll get curd. And then in my experiment, I will test this, whatever I just said, that squeezing lemon juice into milk gives curd. In another case, I can say, milk at 40 degrees Celsius. Take milk at 40 degrees Celsius, add 1 spoon of curd, and then in 6 hours you will get a nicely set curd. And then I will test this hypothesis. So
Hmm.
I am trying to explain something which I am about to investigate.
Okay. So that is the concept of hypothesis.
Right.
Um, in the second activity, you ask students to differentiate between milk and curd without relying on their sense of taste. Why is doing this through other senses important?
The first and most important point is safety. So
Uh,
we always say this, that in the laboratories nothing should be tasted, no matter how clean your apparatus is, or how tempting that food or whatever it feels. Tasting should be avoided because you do not know what chemicals were present before in those beakers or in those tubes or whatever. And
hmm.
another point is, even if someone is doing this activity in the kitchen, when you taste something, it is very easy, or it's very clear, gives you a very clear distinction of what is what. But
Hmm.
when you say you're not allowed to taste, but you have to take a sniff, then it's a very fine distinction to be made. It's like a challenge to your ability to, your olfactory senses. It's very difficult. It's not very easy. Some students can do it very quickly, but for others, you have to take a sniff 2 3 times, and then you can say which is which. So it's sort of a
Hmm.
challenge. And
Hmm.
we are not just again saying you only have to take a sniff. You can also touch. Another
Hmm.
way we figured out is where you can, you can swirl so you can just swirl it like this and see if it is moving, if it is
Hmm.
as fluid as milk.
Hmm.
Excuse me. So that is the other thing can be done.
Okay, so you already answered part of my question about, you suggest that this activity could be used to discuss the many senses and extended tools used for observation in science. What does this mean and why is this important?
So here, by extended tools we mean, what else are students using to understand this process of curd formation. They are not using any high end equipment. They are not using very costly things in to test. At the
Hmm.
most, they'll be using beakers. They'll be checking using their senses, swirling, trying to run it through a slide or something. So these are the, extra things that they are bringing to the bench to better understand this process of curd formation.
Okay. In the third activity, you ask students to set curds themselves. You suggest that this can help them understand the process of standardization in science. What does this mean and why is this important at the school level?
When we talk about standardization to students, we use again in a very simple manner. We tell them that any process that is to be termed as an experiment that works, one has to run a series of experiments to find the best conditions in which that experiment will work. So again, we take the example of scientists when they standardize, when they say that okay, this is an experiment to say determine protein concentration. Someone will have done a series of experiments to check how much protein should be taken, what should be the amount of dye that is to be added, and how to quantify the protein present, to give an example. So
Correct.
when in this case, okay, let me come to this example now. If I want to find out the best condition for curd formation in the least amount of time, I will have to first see I'll first check what is the proportion of milk to curd. So I will take milk at different volumes, say half a cup, 1 cup, 1.5 cup, and so on. I will add the same volume of curd to each tube or each cup.
Hmm.
And then I will keep the entire setup at the same temperature. Okay, so when I'm standardizing in every round of experimentation, I am changing one component only. Towards the end, I will know that okay, for example, to 1 cup of milk, if I add 1 teaspoon of curd, keep it at say 37 degree for 8 hours, then I get curd. So to for me to arrive at this conclusion that okay, these are the conditions which will give you curd in 8 hours, I have done standardization, where at
Hmm.
each step I have decided or I have figured out what is the condition that gives me the desired result.
Hmm.
So standardization is where you do a series of experiments to figure out that best condition. Another thing we can tell students is that for any standardized experiment, whenever another person repeats that experiment, he or she should get the same, or they should get the same outcome.
Hmm.
So if I am working on a standardized experiment of protein, determining protein concentration, anybody
Hmm.
doing that experiment should get the same standard curve as I am getting, or a very similar standard curve. So that
Okay.
is the aim of standardization.
Okay. I'm curious, what conditions of setting do students generally discover in this activity? Are there any points of differences or disagreements, and what do
Like I
they?
said initially, yeah, initially we get very different kinds of responses, which include just keep milk, 1 cup of milk as it is.
Hmm.
Whereas at the end of the activity, students figure out that you can add either a small amount of milk, small amount of curd or buttermilk, or a whole chili with its intact stalk, to milk, and then you will probably get curd.
Hmm.
So initially, they also talk about adding lemon juice or tamarind juice, vinegar, or any acid to milk.
Hmm.
So before they work on the experiment, and later they figure out that only adding curd or the other things like buttermilk or chili can help.
Okay. All right. This activity can help students learn what that the kind of bacteria that cause milk to spoil are different from the kind that cause curd to form. Are there ways in which students can know this for themselves? Would using a simple microscope as an extended tool help?
To answer the first part of your question, students are aware of the fact that spoilage is caused by microorganisms. They
Mhm.
also know, most of them know that you cannot keep milk for a very long period, but you can safely keep curd for a week or say 5 days under refrigeration.
Mhm.
They are aware of the fact that even curd can get spoiled by microbes, although we don't tell them which are those microbes.
Mhm.
um, Talking about the use of a microscope, so some students had asked us if we can show them how these lactic acid bacteria look under the microscope. So if you take a drop of curd or buttermilk, it's very difficult to observe. It's a very hazy thing.
Mhm.
Because I was trained in microbiology, I was able to demonstrate the presence of lactic acid bacteria using a special technique. It is called gram staining.
Oh.
So, gram staining is a very simple technique, and it's also very easy to be learned. And gram
Mhm.
staining kits are available in the market for teachers. So, if
Okay.
anybody can do this technique or can learn it and then demonstrate the presence of lactic acid bacteria, there's nothing like it. It's a very interesting. Students get fascinated by what looking at those bacilli, those rods under the microscope.
Sure.
So we did that when we had a camp at HPCSE.
Okay. Do students also have curiosity about, I mean, do they wonder about how did the first batch of curd get formed? Was it just a happy accident? Do students ask if the chances of such an accident happening again can be improved by playing around with some conditions of storing milk? Do they come up with things like that?
Yes, they do ask this question about who set the first batch of curd, and then yes, we tell them that the legend carries that there was a person who carried milk in a goat skin bag, and the goat skin bag was teeming with lactic acid bacteria probably, and that's how it transformed milk into curd. But I haven't come across a question which said that which a question where someone asked, "Can this happen again?" I
Mhm.
haven't come across that kind of a question.
Okay. And is this true? Is this the truth? This goat skin and legend? You said it's a legend. We don't know.
Yes, most reports say the same thing that legend carries, so we don't know. But it
Let's
appears to be the most plausible
right.
reasoning. Yeah.
Yeah. Rohini, you suggest that curd forming bacteria can inhibit the growth of milk spoiling bacteria. Some children may wonder why curd spoils. How do you respond to such questions?
So in that case, we tell them if they are not aware of spoilage being caused by microbes, we tell them that the way there are bacteria that can spoil milk, there are
Hmm.
other organisms like fungi or molds
Mhm.
that can, or other bacteria that can spoil curd. And if someone has been keenly observing and someone had accidentally kept curd in their fridge for a long time, then one can actually see, red colored uh, growth on the inside the bowl of curd.
Uh-huh.
So I have seen that, and therefore I know. But yeah,
Oh,
it depends.
okay. You also ask students to predict if curd can be turned back into milk. How do students respond to this? Is this question left to, just as a hypothesis, or are there students who are encouraged to test this?
In our experience, all students unanimously said that you can't do that. It's an irreversible reaction.
Uh-huh.
So I don't think students would be thinking about the other direction. Most
Oh,
of them agree that it's an irreversible reaction.
okay. All right. In the fourth activity, students are encouraged to test if they can get a fresh batch of curd without an old batch of curd. And you've already talked about this earlier. When students use lemon or green chilies instead of old curd, they see paneer being formed. Do students test this by senses other than taste?
Um, so again, when they come with this idea that you can simply squeeze lemon juice into milk, they actually are disappointed because they see that immediately, paneer is formed. But
Hmm.
when we explain, we have to tell them that immediately you saw the precipitation of casein, I mean the white solid. So that is
Hmm.
paneer. Now, if you strain it, it is just paneer. Okay, you can feel it with your hands.
Hmm.
So if this was a biological reaction, there are no organisms that are playing anything here.
Hmm.
It's a purely a chemical reaction. You can add acid to milk and still see the same reaction, where you can see
Hmm.
the white precipitate being formed. Whereas in
Hmm.
curd formation, it is microbes in action. The
Hmm.
bacteria are actually fermenting lactose. So it's going to
Hmm.
be a slow process. And that's how
Hmm.
you can differentiate between the chemical process of paneer formation versus the biochemical process of curd
Hmm.
formation. So we have to tell them in the
Okay.
class.
All right. You suggest that this activity can be used to introduce a discussion on the use of controls in scientific experiments. Could you please elaborate, Rohini?
Again, in simple terms, in my experiment, a control will be something that will not change, uh,
Hmm.
or nothing will change in that control over time
Hmm.
with along with the experiment. Or, in other words, a control is something which will give me either it will definitely give me what I'm looking for, or it will definitely not give me any change.
Hmm.
So, for example, when I am looking at curd formation as an experiment, I will
Hmm.
just keep one tube or one cup with plain milk.
Ha.
I will not add anything to that milk, and that milk will be at the same temperature as my experimental tube milk, as in the milk in my experimental tube. So
Hmm.
suppose I'm monitoring my tubes at every hour or every 2 hours, I will also take a look at what is happening to this plain milk tube.
Hmm.
So that will be my control. I'll compare my experimental setup with the
Hmm.
control. I can also keep another control where I have freshly prepared curd.
Hmm.
So this is how freshly prepared curd looks. This is how milk looks. Now,
Hmm.
how are my tubes doing in comparison to these two tubes? So
Hmm.
that is how we again we it takes a while to for students to process this, but then
Hmm.
it's
Correct.
important when you are doing, you have to compare it with something that is for sure will give you the result or will not give you the result.
Hmm. Okay. You also mentioned how you use this activity to introduce students to the difference between the purely chemical change in paneer formation versus the biological change in curd formation. Why is this important? I mean, again, are there some ways in which students can know this for sure? This for themselves, in fact.
In our trials, I don't remember students saying this that they knew.
Hmm.
Only after performing the experiments they get to know.
Hmm.
If there's someone who has helped in paneer formation in the kitchen, then that student might know. But otherwise, they most students figure out at the end of the experiment
Okay.
about
So
the
they.
chemical process, the biological change.
Okay. Okay. This entire activity is such a lovely way of introducing students to how one can tell pseudoscience and science apart. Do you explicitly discuss this aspect in class? Any suggestions
Uh,
for teachers who want to facilitate such a discussion?
we don't explicitly say this,
Oh.
but I must say that this activity
Hmm.
can be seen as a stepping stone in developing scientific thinking. Because the
Hmm.
students are, we are asking so many questions, students are made to ask a lot of questions. That is how they'll develop scientific thinking. And this process has to continue all their lives. It's a lifelong process. And they should ask a lot of questions in different contexts, not just in the case of this activity. So as they learn the way of scientific thinking, they will probably be able to identify pseudoscience and also be
Hmm.
able to give explanations of why something is unscientific. But
Hmm.
here we don't focus on that because we don't want to overwhelm the students. But yes, it can be a very good starting point.
Okay, that's good. In the last activity, students are invited to test the relationship between temperature and the process of curd formation. You use this experiment to introduce students to the concept of variables and the necessity of changing one variable at a time. Could you elaborate on this?
Again, we explain variables as that component which I am investigating in my experiment or in this series of my experiment. That is something I am investigating. So when I say what is the volume of curd that is to be added, so volume of curd becomes my variable. I will
Hmm.
change the volume of curd for every single tube that is a part of my experiment. If I am saying the temperature of milk to start with is what I'm trying to test, then I will take milk at different temperatures, 30, 35, 40, 45, and so on. So in this case, temperature becomes my variable. So what is it that you are testing should become the variable, and rest of the things should remain constant.
Okay, all right. You ask students if they can arrive at the temperature range at which curd formation is fastest. Is this question aimed at getting students to, find out for themselves, or to offer informed guesses, or to encourage students to discover the limits of this range through experimentations?
So when we try this in schools, once the students have set the experiment, it's under incubation. We have a lot of time. We get around 2 hours. What do you do
Mhm.
in 2 hours? So we make students predict the outcomes, and then they
Mhm.
discuss with each other. Okay, what will happen in which tube and what should not happen. So yes, guessing is something we are allowing in this case, or we want students to do that. We want them to predict. Uh, we are not explicitly using the term limits of range, but
Mhm.
because many students are doing such an experiment for the first time, we should expect a lot of variation, hand to hand variation, or what someone calls warm for the other person, it might be hot. So we have to give them this range instead of
Mhm.
giving specific a specific number, which is difficult to arrive at the stage where they are right now. So
Okay.
that's the reason we say, okay, tell us a range roughly.
All right. You end the article by suggesting that although the article provides experimental designs, it may be best to encourage students to come up with their own experimental designs whenever possible. Why is this important, Rohini, and what do students learn from it?
When we allow students to design their own experiment using their own their own understanding, they look at the experiment as their as their own brainchild, or it can be said that they take ownership of their experiment. And
Hmm.
that is one big thing, because they want to constantly keep observing, They want to know what is going to be the end result, and they also
Hmm.
want to share it. So when
Hmm.
they take ownership of something, I think that itself is a big achievement for them. And
Correct.
then it follows, like they would want to try out different things once they have done one experiment on their own. They would
Hmm.
definitely want to continue that.
Okay. So that is why. Depending on the school, some students may be hesitant or not, know how to design scientifically valid experiments. Do you suggest giving students some kind of introduction?
Yes, an introduction is important, and yes, students do hesitate, and that's the reason we initially engage them in groups rather than
Hmm.
asking them to do it on their own. So, in within a group, students like exchanging ideas. There is peer involvement. Uh, at times, we may have to give or offer some help. We may have to give initial guidance, and then they can continue. That also works well with students.
Okay. Okay. And once students design their own experiments, they may ask the teacher for feedback. Would you have any guidance for how teachers can respond to such requests?
So I think it depends on the kind of questions that are coming to the teachers, and most teachers are aware of this process of code formation. And both
Hmm.
in our resource as well as the article, we have provided information for teachers.
Hmm.
So that is how teachers can take a look at the guide, at the what we have provided, the information that we have provided. There are a lot of articles around this process again,
Hmm.
which teachers can look at. So I think the internet is again a very good resource to go to.
Hmm. Hmm.
And teachers use their experiences.
Hmm.
This we have seen this in teacher camps where they tell us, no, this is what will work and this will not work. So they
Hmm.
know a lot from their own experiences of code formation.
Correct. Correct. Okay. You suggest that milk and curd can be effective as live educational tools. What does this mean and why are they important?
In this case, it's microbes in action. Like I said, the microbes, that the lactic acid bacteria are constantly going to ferment lactose, and we are going to although we are not going to look at the organisms in real time, but we can see the change they are producing. We can
Hmm.
take a sniff, we can see, okay, curd is for are they forming curd or are they not doing it, or is there no change? So we are
Hmm.
able to understand indirectly that, okay, they are growing and they are
Hmm.
producing something. So that is the live organisms, and we are looking at them, I mean looking at them indirectly. That is what we mean here by live educational tools, where something
Oh.
is in action, you're not stopping it, but you
Hmm.
can stop it. So knowing that and playing around with it
Hmm.
is something very interesting.
Okay, okay. Rohini, that's a lot of information coming from you, and I'm, so glad that we were able to ask you all these questions. Is there anything you'd like to add to this?
Well, it was quite an experience, quite an enriching experience. I also got to learn a lot while developing this module, writing the article, and also in the school trials. So it was a very, very enriching experience for me as well. And we also got to know how many things can be done, how many kitchen tools can actually be used as educational resources. I would also like to take the opportunity to thank two more people. One is Dr. Ritesh Kunyakari from the Tata Institute of Social Sciences. He's an associate professor, and he had helped in shaping this entire module as well as writing the article. I
Oh,
also want to thank Chitra Ravi, who is in the editorial team of iWonder magazine, and she has also helped immensely in designing the activity sheets which are a part of this article. So thank you, Ritesh and Chitra, for this.
okay. Rohini, so thank you very much. And to all of you who are watching this, thank you for joining us today. And now, dear viewers, here's a question for you. Remember, we asked a question in the last webinar too. Rohini used activities around curd setting to help children learn important concepts in biology, physics, and chemistry in the curriculum. What everyday practice have you used to teach science? Do share a brief note about your approach and experience. Send in your responses or response to this question to iwonder at apu.edu.in. Your response can be between 300 and 900 words long. This is important. Please use snippet on the Milky Way to learn biology in your email, and send it to us. If it is a good fit for iWonder, it will be published in an upcoming issue. Also, if you have any questions for Rohini, please send them to the same email ID, iwonder at apu.edu.in. We will do our best to get them answered. Join us again for our next webinar on October 11th, 2023. And for those of you who have not yet subscribed to iWonder, we do hope you do so now. So, thank you and join us again next month. Thank you very much.